Hi there, science fans. Welcome to another episode of Science and Other Stuff for Senior Citizens. My name is Dan Casciano. If you like these COVID-19 videos, please subscribe to my channel and ring the bell to be informed of future videos. In the last several videos, I described recent advances and the status of 4 vaccines being developed to eliminate COVID-19. In the present video, I will describe recent advances and updates to a variety of experimental drugs or biologics being considered or already in use to treat individuals infected with COVID-19. Here are several experimental treatments presently being used to treat hospitalized COVID-19 patients. These treatments may be given to the patient either during the early stages of the infection or at later stages when the patient is experiencing severe outcomes. You may recognize some of these treatments since they were given to President Trump during his infection by SARS-CoV-2. I will describe each of these treatment types in the following slides. Panel A on the left describes the SARS-CoV-2 virus that contains an RNA genome that provides all of the instructions needed to infect a human cell and produce new viruses. It is studded with the so-called spike proteins that the virus uses to penetrate the host cell, in this case a human respiratory cell. The virus enters the host cell shedding its outer coat. The RNA genome hijacks the host protein synthesizing machinery and makes the replication transcription machinery depicted as bluish-purple in the lower left-hand portion of diagram A. The replication machinery then begins making new viral RNA as shown in the boxed-in section of diagram A. As new viral RNA instructional molecules are made, they are then inserted into a new virus that then migrates to the cell membrane to infect other cells and hosts. As shown in diagram B, this replication process is inhibited if the host is treated with a nucleotide analog inhibitor like remdesivir. The viral RNA cannot make new instructional molecules, and thus the infection process is inhibited, and the infected cell and the virus is killed. If the human host is treated early in the infectious process, and if the human is treated with supplementary medications like monoclonal antibodies and anti-inflammatory drugs, the patient has an excellent chance of surviving COVID-19 infection. This cartoon depicts how the SARS-CoV-2 infected cell can respond to the infection. Step 1 shows that upon infection, the virus is sensed within the cell and begins to produce signaling molecules called cytokines. These cytokines recruit a myriad of different immune cells to the infected cells and proceed to either kill the infected cells or produce specific antibodies to kill the virus that has invaded the body. The recruited cells in turn send regulatory signal molecules to their neighbors indicating enough cells have been called to defend against the virus and are told to stop replicating and return to the resting state. This is depicted in step 2 of the process. The result is shown in the section of the diagram where a functional immune response is shown. However, if in step 2 an excessive number of immune cells are attracted to lung cells and the regulation of their function has gone awry, then the cytokine storm results and a dysfunctional immune response occurs. If this systemic cytochrome storm is not abated, then the result is multi-organ damage. This is depicted in step 3b. If the patient cannot control a dysfunctional immune response, steroid drugs like dexamethasone are given to the patient to reduce the cytokine inflammatory response. This slide shows another outcome of dysregulated cytokine storms. Section 1 of this diagram lists some of the immune cells recruited by the intracellular sensors that recognize a foreign invasion. Section 2 indicates some of the cytokines produced by these cells to defend against foreign invasion. If there is an overproduction of certain of these cytokines, specific clinical outcomes are identified of which some could be lethal. One such clinical outcome is disseminated intravascular coagulation or blood clots. Blood clots can lead to cardiovascular events like stroke or heart attack. Treatment with anticoagulants like heparin sulfate can mitigate the clots and reduce significantly multi-organ damage. Monoclonal antibodies, like the ones developed by Regeneron and given to President Trump, are produced in several ways, two of which are presented in this slide. Diagram A on the left uses mice that are challenged with an antigen, in this case the spike protein of SARS-CoV-2. After a period of time, spleen cells containing the activated immune cells responsible for the synthesis of antibody to SARS-CoV-2 are harvested and in vitro cultured with special myeloma cells. Special techniques are used to cause fusion of the two cell types and are placed in a specific growth medium that allows selection of the desired fused cells, called hybridomas. These hybridoma cells are further cultured, allowing the increase in numbers of this cell type, as well as the production and secretion of that antibody that was developed specifically to inactivate SARS-CoV-2 virus. This monoclonal antibody is then injected into an infected patient to help in neutralizing the free virus in the blood and organs. A second method is to use blood from a person who has recovered from COVID-19. This is shown in the B section of the diagram. The blood contains immune cells that have the ability to manufacture antibodies against SARS-CoV-2. These cells can be isolated from the blood, and their DNA can be isolated in the laboratory. The part of the DNA that instructs the production of the SARS antibody can be isolated and introduced into specialized cells that can act as factories for the mass production of SARS-CoV-2 specific monoclonal antibodies. Use of monoclonal antibodies have several limitations, including short-lived protection from the virus, and they are extremely difficult and expensive to produce. Drugs that are shown to be safe and efficacious for other diseases are being evaluated for potential use in decreasing the severity of COVID-19 infection are shown in this slide. Two are highlighted, namely hydroxychloroquine and melatonin. Human trials of hydroxychloroquine, a drug found to be useful in some immune deficiency disorders, was shown to be inactive against SARS-CoV-2 and in some cases caused damage to some individuals in the trial. Melatonin, a hormone produced by the pineal gland and used as a sleep aid, especially for insomniacs, was found to have potential by researchers at the Cleveland Clinic's Lerner Research Institute. These scientists ran a computer analysis to identify drugs that could possibly be repurposed to treat COVID-19, and melatonin emerged as a potential candidate. These researchers are suggesting experimental examination of melatonin to treat COVID-19. Although vaccination is imminent, we must continue to clean or wash our hands often, avoid close contact with sick people, practice distancing 6 to 10 feet, avoid indoor and outdoor crowds, and wear a mask properly. We will continue following these CDC guidelines until we reach herd immunity. For more information about science, visit my website at www.dcasciano.com. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comment section below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and ring the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye.